Okay, hello everyone. I'm Shamim, a PhD student from Goethe University Frankfurt. I would like to present you Gaze Shiny, a comprehensive and interactive visualization of transcriptional regulation in single cell resolution. All we know, gene regulation in single cell is very important. So in this work, we would like to understand gene regulation in single cell data and visualize it in a simple and interactive way. And TFs are a key player in gene regulation. There are different assays to investigate genes like single cell RNAs, single cell A tag, but there is no assay to infer TF activity at single cell level. So we need to do some computational methods to infer TF activity at single cell level. Okay, so we have developed a statistical framework named Gaze to infer TF activity per cell. And also we developed a Shiny app named Gaze Shiny to visualize the output of our Gaze pipeline. The input data could be single cell RNA or both single cell RNA and single cell A tag. And after doing our computational methods and steps, we get Gaze object and send it to the Gaze Shiny app to see the result. Here, I would like to mention some similar apps, some recent ones. The first one is Shiny Multiome.uio, which published in 2023. The input data is single cell RNA or both single cell RNA and single cell A tag, and it allows users to see transcription factor footprinting, UMAP, and single cell A tag peaks. Two other ones, SCUBI and SC. The input data for both is single cell RNA, and they allow user to actually explore the data and see some plots like UMAP and gene expression. But a unique feature of Gaze Shiny is a visualization of a TF gene regulation on single cell level. Before showing the app, I would like to shortly explain our Gaze pipeline. As I mentioned, our data is single cell RNA and single cell A tag. We kind of join these, integrate these two data sets to a joined space, and then we create meta cells. To make all of you know, single cell data is a sparse data. So to actually magnify it, we create meta cells. To create meta cell, we apply a clustering and on a cells, and then we get the average of each cluster as a new cell or meta cell. And then we use STAIR, which is a tool developed by my colleague Dennis to get enhancer gene leaks per meta cell. As you can see, the matrices, for each meta cell, we have a matrix, which is genes times TFs. And then over the 4 steps, we compute TF gene score per meta cell, and we learn a TF gene regulation, and finally estimate TF activities per meta cell. I'm going to explain these 4 steps in the next slides. Okay, so as I mentioned, we got these matrices from STAIR, which is genes times TFs per meta cell. And then we kind of reshape these matrices to get a matrix of per gene, a matrix of meta cells times TFs per each gene. So as you can see, we have these green matrices, matrix, which is meta cells times TFs. We got it from STAIR. And also we have the red one, which is gene expression in meta cells. So by running a regression model, we can get the purple vector, which is TF activity. So this is for one gene, and we did like this regression model for all of our genes. So it's a per gene model. And by using explainable AI, we actually get the score of each TF For example, in here for gene 1, TFM is the most important one, the highest score, and TF1 is the lowest score. Okay, so now I'm going to show you some screenshot of our Gaze Shiny. Our app has some tabs. The first one is data exploration. And here I'm going to show you Lucas Nissin dataset, which is single cell RNA for healthy and heart disease. And it has 16K genes. Okay, in the left panel, users can select desired gene. For example, here got a 4. And users also can select mode single cell or meta cell or both. And if there is condition like healthy or a specific disease or range of age, users can select and see the result. Here I'm showing in the right a UMAP of selected gene, and also in the below gene expression in different meta cells for the selected gene. Also, users can see how a gene differentially expressed in 2 different conditions, for example, a healthy and a disease. If users select more than 1 gene, they can see the average read counts for selected genes in different meta cells. Top one is in meta cell level, and the below one is for single cell level. Also, users can upload their desired gene as a TXT file and see the result. The next tab is differential analysis tab. Here I'm showing results of MPAL dataset, which is single cell RNA and single cell ATAC, and it has 16K genes. And also we have 2 views in the left, TF view and gene view. Here I'm showing gene view. Yeah, here users by selecting the gene view can see all genes and or differential test. So here I'm showing one of those selected genes, CD74, into specific cell type here, B cell and HSC cell. The orange points are mean SHAP values, and as you can appreciate, FOSL2 has the highest rank, or is the most important TF in B cell for this gene. And also in the table, we showed the top ranked TFs and their p-value based on our statistical test. Okay, as a summary, Shiny is a flexible app which allows users, especially who have limited programming knowledge, to explore the data and understand the data. And a unique feature of Shiny app is visualization of TF gene regulation. The input data could be single cell RNA or both single cell RNA and single cell ATAC, and both or Gaze pipeline and or app are implemented in R. Yeah, I would like to thank my supervisor, Professor Schultz, and my colleagues who have been working on this project, and thank you for your attention.
Oh, thank you. Do we have questions, or is everybody too hungry and wants to get to the lunch break? Well, I actually have one, more of a technical question. So the design of the interface of your application, was that just so to say what you thought would make sense, or was that designed together with actual users? The user interface, the design of that, was that designed together with the users, or is that what you think would make sense? So did you so to say make a requirement analysis with the users, or so to say are you the user of your own system? So how did users so to say react to that? So
you know, we haven't uploaded it publicly. It's under test, but users can easily. There's a help section. Users can read the help and easily work with our app.
Okay, right. More questions? Ah, yeah, we have 2.
Thank you for your presentation. I have a question about how do you infer the link between the gene and the transcription factor. you say that you did it at metacell level, but is it possible to do that at single cell level if you have only a data set of single cell and not single
Actually,
cell and ATAC-seq?
And metacell is, the new name for, I would say, single cell. It's not just bulk data, it's single cell. But to magnify the single cell, we change it to metacell. Actually, I explained, and we apply the clustering on cell types and then get the average of them. So it's kind of single cell, not bulk cell. I mean, bulk data. And we get the data you mentioned from STER, the links between TFs and cells, metacells.
So metacell can be, sorry. So metacell can be a single, a cluster of cells then.
Yes.